right, great.
You can just hit F5. Oh, okay. Okay, you got it.
All right, so I think I'll get started. Can all of you hear me? All right, so I'm going to get started. My name is Salila Kerr. I'm the Section Director for Endocrinology. And I'll be talking about the introduction to endocrinology. Obviously, it's just an introduction, so you don't have to know all the stuff in it, but it's supposed to get you excited to start endocrine, which I know is your last block. So today I'll be going over the definition. Today I'll be going over the definition. Is that better? Who said they couldn't hear me? Well, whoever it was, can you hear me now? I sound like the Verizon commercial. Let me put this on. Can you hear me with this? Is there somebody here from... Yeah, no, I got the turn it on concept. I think it is on, but is somebody from AV here? Can you hear me now? All right, let's go then. I know it's an exciting beginning to endocrinology. So today I'll be talking about the definition and principles of endocrinology, and then I'll give you a course overview, and then I'll talk about hormones, the types, their synthesis and processing, hormone action through receptors, regulation and feedback, and then I'll move on to the pathologic mechanisms of endocrine disease, and finally end with the reason we're all here, which is the clinical relevance of this. So just to start with the definition and principles of endocrinology, endo means inside and crine means to secrete. So it's defined as a study of glands and the hormones they produce internally. A hormeo means I set into motion or I stir up. And the definition of a hormone is a chemical substance secreted into the circulation and delivered to target tissues where they produce physiologic processes and are regulated by feedback mechanisms. So these are the different types of signaling. So this is endocrine signaling, where you have the signaling cell secreting, in this case, hormone into the circulation, which then goes and binds to the target cell. So you could think of it as a thyroid hormone. This is a thyroid secreting thyroid hormone into the circulation, and then going to different target cells, such as muscle or the heart. Paracrine signaling is when the signaling cell is close to the target cell, and then they secrete the chemical substance. Autocrine signaling is when a cell secretes its own signal, and justacrine is when, as you can guess, they're close together. So the endocrine system is responsible for homeostasis, in particular growth, development, reproduction, blood pressure, the concentration of ions in the blood, and even behavior, all regulated by the endocrine system. And it's the secretion of hormones and the action at their target tissues. Unlike many of the other courses you may have just finished, it's not defined by anatomy, because as I'll show you in the coming slides, the endocrine organs are all over the body. And endocrine diseases tend to be multisystemic in their presentation, and focusing on one predominant symptom may lead to failure in recognizing the nature of disease. For example, many endocrine diseases present with polyuria, which is urinating too much, and that can be a symptom of hypercalcemia. It can be a symptom of diabetes mellitus or diabetes insipidus also. So these are the main organs we'll be talking about: the hypothalamus and the pituitary is the master gland, the thyroid, and then behind the thyroid there are 4 parathyroid glands, the pancreas, the adrenal, and the gonads, and in men those are the testes, and in women the ovaries. So just to go over the course There's lectures and small groups, and every year in the comments people write, oh, I was surprised that our small group leaders knew our name. I already sent them everybody's picture, and they probably will know your name the first day. And we do make it a point to try to learn all your names. We used to get bios, so I used to scare people by being like, oh, how was undergrad there? But we don't get that anymore. So if you want to tell me, I'll try to remember. And we do have 1 to 2 cases per session. And please read the cases ahead of time. It's a little bit of a waste of time to show up if you haven't read the cases. We only have 2 cases usually per session, and the preceptors really prepare them ahead of time, so it would be good if you could too. And there are 3 TBLs, and they're also labs. And the first TBL is this Friday. You want to come up here and say that? No? So the recommended texts are Physiology by Costanza. And then in the past few years, some of the students have told me they've been using Endocrine Pathophysiology by, I cannot say her name, but that's also a good book to use. But really, the main required text is Physiology by Costanza. So we'll be going over, this is the way the course will be set up. We'll be going over pituitary and growth, then thyroid, then adrenal, male reproduction, and female reproduction. And much of the female reproduction section is taught by OB/GYN, whereas the vast majority of the rest of the lectures are taught by the endocrine faculty. And in particular, in female reproduction, we'll go over pregnancy, labor and delivery, contraception, fertility, and acne and hirsutism. And then we'll have calcium and bone metabolism, and then diabetes and obesity. So I did make a little bit of a change to the curriculum this year, and there'll be a little bit more obesity slides, since obesity is probably the biggest endocrine disease we have. And just to go on to talk about hormones, their type, and synthesis and processing. So these are the endocrine glands. And the hypothalamus makes hormones, or releasing hormones that trigger the anterior pituitary, which then work at target tissues. So for example, TRH stimulates TSH by the anterior pituitary, which is thyroid stimulating hormone. And as its name implies, it works at the thyroid to make T3 and T4, and T3 is the more active form of that hormone. And then they go to their target tissues, such as muscle, the heart. And the hypothalamus also secretes GnRH, which works at FSH and LH, and that works in the testes or ovaries, depending on whether you're a man or a woman, to cause the intended response, and so on. And then there are a few hormones made by the hypothalamus, such as dopamine, which actually inhibits prolactin secretion. And Dr. Wardlow will talk about that next in her next lecture. And then there are other hormones that are completely independent of the hypothalamus and pituitary axis, such as insulin, which is made when you eat and have glucose, and parathyroid hormone, which is stimulated by low calcium levels. So the most common form of the hormones are peptide or protein hormones, which are synthesized from amino acids. So the peptide hormones, as you can see, because they're boxed in here in red, are the vast majority of hormones. So most of the hypothalamic hormones, excluding dopamine, all of the anterior and posterior pituitary hormones, parathyroid hormone, calcitonin, the hormones of the pancreas, and renin, which is made by the kidney, which you guys are probably very familiar with after your last block. So the majority of the hormones are peptides, and these are just 2 examples. So parathyroid hormone is one, and I'll go over what parathyroid hormone does in a little bit more detail. And then proinsulin is another, and proinsulin is cleaved to connecting peptide, which is C-peptide, which we use clinically to see if a patient's making their own insulin. Because if you're making insulin, you also have to have C-peptide in your blood. And it's one of the ways we use to distinguish whether a patient has insulin-deficient diabetes or type 2 diabetes, where you actually have high insulin levels usually. So just to go over peptide and protein synthesis in more detail, in the nucleus, the gene for the hormone is transcribed into an mRNA. The mRNA is transferred to the cytoplasm and translocated on the ribosome to the first protein product, which is pre-pro hormone. And translation then continues on the endoplasmic reticulum until the entire peptide sequence is produced. And the signal peptide is removed in the endoplasmic reticulum, converting the pre-pro hormone to a pro hormone. And the pro hormone contains the complete hormone sequence plus other peptide sequences, which will then be removed at the final step. And then the pro hormone is transferred to the Golgi apparatus, where it's packaged in secretory vesicles. Proteolytic enzymes cleave peptide sequences from the pro hormone to produce the final hormone. So the final hormone is stored in secretory vesicles until the endocrine cell is stimulated. So for example, parathyroid hormone is a peptide hormone. It's synthesized and stored in vesicles in the chief cells of the parathyroid glands. So again, the parathyroid glands are the 4 glands behind your thyroid. And the stimulus for PTH, or parathyroid hormone secretion, is low extracellular calcium. So if your calcium is low, you have increased production of parathyroid hormone, which then goes to the bone and breaks down the bone to release calcium into the circulation. And it also has other effects, which we'll learn about in some of the coming lectures. And when sensors on the parathyroid gland detect a low extracellular calcium concentration, the secretory vesicles are translocated to the cell membrane, and PTH is released into the blood by exocytosis. So this is actually probably the most famous case of hyperparathyroidism, and I'll just go over a little bit about what happened to this man. So this is the same person, and this is Captain Charles Martel. And he had hyperparathyroidism. So when you have hyperparathyroidism, the parathyroids function independent of the signaling. So even though the body has high calcium levels, their parathyroid gland is adenomatous and is enlarged and secreting parathyroid hormone, which then works to break down bone. And this is actually what happened to this sailor. So he was 6'1" and he became 5'6". And this actually happened in the 1920s. And so I told you there are 4 parathyroid glands in your neck. So he had 6 operations, a couple of them here at Columbia, but we won't bring that up. And then he went up to Boston, and they still couldn't find where, and this was at Harvard that I will bring up, and they still couldn't find his parathyroid. So he actually went to the Harvard Medical School library and figured out there's such a thing as an ectopic parathyroid gland that's in your chest, and that's where his was. But unfortunately, 2 weeks after they found that parathyroid gland, he actually died from complications of hypocalcemia. So this is just a case to illustrate what uncontrolled endocrine diseases can do. And luckily, since the advent of the Chem-8 in the 70s, people are catching hyperparathyroidism way before it becomes symptomatic. And now it's really an asymptomatic disease that we're finding just on routine labs. So the second type of hormone are steroids, which are derivatives of cholesterol. So these are the steroid hormones, where all of the hormones of the adrenal gland, so cortisol, aldosterone, and androgens, as well as the reproductive hormones of testosterone, estradiol, and progesterone, and then the hormones of the corpus luteum, which are estradiol and progesterone, and then 125D, which is made by the kidney. So steroid hormones are synthesized and secreted by the adrenal cortex, the gonads, the corpus luteum, and the placenta. And all steroid hormones are derivatives of cholesterol. And this is the chart that's probably an endocrine fellow's nightmare. But it starts with cholesterol, and then you do a series of enzymatic reactions, end up with aldosterone, which is a potent mineralocorticoid, which is made by the adrenal cortex, the glomerulosa, and the cortisol, which is again made in the adrenal gland at the level of the fasciculata, and then the DHEA and androstenedione, which are the androgens made by the adrenal gland. So steroid hormone synthesis happens when cholesterol is modified by either removal of a side chain, addition of a side chain, hydroxylation, aromatization, and steroid hormones diffuse into the circulation as they are synthesized, and secretory rates correlate with the rate of synthesis. And again, these are just to bring you back to orgo, some of the chemical structures of the adrenocortical steroids. And lastly, there are the hormones that are derived from amines, which are derivatives of tyrosine. So as you can see, there are very few of these. There's dopamine, which is a catecholamine, norepinephrine, epinephrine made by the adrenal medulla, and T3 and T4, which are made by the thyroid gland. So as I said, the catecholamines are amine hormones, as are thyroid hormone, and they're all derived from tyrosine. So this just shows catecholamine synthesis, where you start with tyrosine, and through a series of reactions, you end up with dopamine, norepinephrine, and epinephrine. And thyroid hormone, and one of the reasons I wanted to illustrate this is really the only difference between T4 and T3 is that T4 has 4 iodine molecules and T3 has 3 iodine molecules. So although they're both active, T3 is the much more active form of thyroid hormone. So the different hormones have different half-lives in circulation. T4, the circulation half-life is about 7 days. And glucocorticoids, their half-life varies. Whereas protein hormones or peptide hormones have short half-lives, usually less than 20 minutes. And we can take advantage of this clinically because these hormones are rapidly degraded. For example, some of you may see if you're on the endocrine surgery service as in your clinical year, that when you remove a parathyroid adenoma, you check the PTH level at time 0, and then you check it after you take out the gland. And if it doesn't drop by 50%, you haven't found the right gland. And you may find this out the hard way because when I walk to my office, I always see med students running to the lab to get this done. So they do it 0, 15, 30 minutes, 40 minutes, and check to see that the parathyroid level has dropped. So we take advantage of the fact that this hormone has such a short half-life to determine whether or not you've taken out the right gland that's causing all the symptoms. So many hormones circulate in association with serum binding globulins, T4 and T3 with thyroid binding globulin, cortisol to cortisol binding globulin, or CBG, androgen and estrogen with sex hormone binding globulin, or SHBG, and calcium circulates bound to albumin. And it's often the pre-portion that's physiologically active. So now to move on to hormone action through receptors. So they can work through membrane receptors, which is what peptide hormones and catecholamines do, or they can work through nuclear receptors, which are small molecules that can diffuse across the cell, and these are thyroid hormone, steroids, and vitamin D. So many of these hormone actions are through G-protein coupled receptors. So G-proteins are a family of membrane-bound proteins that couple hormone receptors to effector enzymes. And G-proteins serve as molecular switches that decide whether the hormone action can proceed. So here is a lovely rendition of a G-protein. So this is a transmembrane receptor, and this is a heterotrimeric G-protein with the alpha, beta, and gamma subunit. And when it's bound to GDP, it's inactive. So when the hormone binds to the receptor, the alpha subunit releases GDP and binds GTP, and which then frees the alpha subunit from the other two subunits. And then I couldn't actually figure out how to make it migrate through the cell, but you get the idea. The arrow means it's moving. And it goes to the adenylyl cyclase and converts ATP to CAMP, which then has the downstream effects that lead to the hormone's actions. So there are three major mechanisms of hormone actions on the target cell. So one is the adenylyl cyclase mechanism, which you see AMP as a second messenger. The phospholipase C mechanism, which uses IP3 and calcium as the second messengers. And then steroid hormone mechanism as well. In addition, you also have insulin and insulin-like growth factors, which act on their target cells through a tyrosine kinase mechanism. And several hormones activate guanylate cyclase, in which cyclic GMP is the second messenger. So this just goes over which mechanisms these different hormones use. So as you can see, a lot of the pituitary hormones use the CAMP as their second messenger, whereas many of the hypothalamic hormones use the phospholipase C. And as the name would imply, many of the steroids, including estrogen, progesterone, testosterone, aldosterone, they use a steroid hormone mechanism. So now this is the part of endocrine that I find most interesting, which is the regulation and feedback. So endocrine hormones maintain homeostasis. And to do this, you need adjustments in secretory rates. So some of these hormones, such as catecholamines, use neuronal mechanisms. So you have preganglionic sympathetic nerves that synapse on the adrenal medulla, and when stimulated, cause secretion of catecholamines into the circulation. And then you also have feedback mechanisms, so positive and negative feedback. And negative feedback is the most important and most common mechanism. So negative feedback, there's some feature of hormone action directly or indirectly inhibiting further secretion of the hormone. And positive feedback is uncommon, and some feature of hormone action causes more secretion of the hormone. So this is negative feedback. And actually, when I was a medical student here, I used to sit actually right over there. And we used to transcribe lectures because we didn't have podcasts. So I was actually assigned to transcribe the endocrine lecture. And I remember looking at the negative feedback and thinking, these people are crazy. Why would they do this? It's so complicated. But it actually isn't that complicated, and I end up going into it. So don't be overwhelmed by all the feedback. I mean, really, the arrows, well, they go in both directions, but I'll talk about why. So the hormone levels, what negative feedback is when hormone levels are judged to be adequate or high, further secretion is inhibited. And when hormone levels are judged to be inadequate or low, secretion of the hormone is stimulated. So here are some examples. So the hypothalamus stimulates the anterior pituitary. So the hypothalamus in this feedback system, which is the testicular one, the hypothalamus makes GnRH, which works at the pituitary to make LH and FSH, which then work at the endocrine gland glands to make testosterone. And when you have testosterone, it goes to the muscle and has its targeted effects and goes to other places also to its target tissues. But once the level of testosterone is high enough, it goes back to the anterior pituitary and shuts down FSH and LH production. And it also goes back to the hypothalamus and decreases GnRH production. So it basically shuts itself off when the levels get high. And you can substitute any hormone here. You can put in thyroid hormone or cortisol. They all work the same way by going back to the anterior pituitary and hypothalamus and shutting down the production when levels are high enough. And there's also a short loop where once the anterior pituitary levels are going or getting higher, they also go back to the hypothalamus and shut down production of the hormones that stimulate their own release. And this is also independent of the hypothalamic pituitary axis, because you can see this with insulin. And no one's having a bagel here today, but usually there's someone having a bagel. So if you eat a bagel, your glucose levels go up, your insulin levels go up. Oh, you're eating a bagel. There you go. Your insulin levels go up when you have glucose, and then that drives your glucose levels down. As soon as your glucose levels go down, your insulin levels also go down. So that's also negative feedback. So the entire endocrine system is mediated by either negative or positive feedback. The vast majority of hormones are regulated by negative feedback. And if you understand this concept, you'll understand most of endocrinology. So there's also positive feedback. So really the main time that this is important is during the menstrual cycle. So during the follicular phase of the menstrual cycle, the ovaries secrete estrogen. And the estrogen acts on the anterior pituitary to produce a rapid burst of FSH and LH secretion. And FSH and LH cause stimulation of estrogen secretion. So essentially what you have is when the estrogen gets high enough, it goes back to the level of the anterior pituitary and hypothalamus and causes LH release. And this is actually used clinically. If you buy any over-the-counter ovulation monitor, that's what they're measuring for, is this LH surge. Because if you don't have the estrogen feedback, the positive feedback to cause this LH surge, you won't ovulate. And the monitors use that to determine when a woman is ovulating to help her get pregnant. So all of these are very clinically relevant. So there are other things that regulate the endocrine system, in particular seasonal changes, light and dark cycles, sleep, meals, and stress. And one of the cases that we go through is how stress can turn off the menstrual cycle, and it works through this mechanism of turning off FSH and LH production. So the endocrine systems that are most regulated rhythmically are the menstrual cycle, the AM peak of ACTH and cortisol. Peptide hormones are secreted in discrete bursts. And there's a pulsatile nature and a rhythmic pattern that's important, for example, in the GnRH pulse frequency. And this just shows that if the GnRH is not secreted in a pulsatile fashion, you don't get these LH pulses. And if you don't get an LH pulse, you don't ovulate. So it's very important. And they're actually developing now a GnRH pulse generator that you can wear that'll stimulate your body to make LH in a pulsatile manner so that you can ovulate. And then this is something that we take advantage of clinically, which is that cortisol also has a burst. So in theory, if you sleep from midnight to 8 o'clock, at 8 a.m. when you wake up, your cortisol should be at its highest level. And at night, right before you go to bed, around midnight, it should be at its lowest level. So we take advantage of this clinically. If we think a patient is not making enough cortisol, we have them check it at 8 a.m. when it's supposed to be at its highest. And if it's still low then that indicates to us that the patient may not be making enough cortisol. Whereas if we think the patient's making too much cortisol or has Cushing syndrome, we check their levels at midnight when they're supposed to be low. And if they're still high then there's a higher suspicion that they may have inappropriately high cortisol levels at nighttime. So then I wanted to move on to the pathologic mechanisms of endocrine disease. So the main things are you have hormonal excess, hormonal deficiency, or hormone resistance. And this is a patient with hormone excess. This is Graves' disease. And the things I wanted to point out here are that she has a very large thyroid gland. She has a goiter. She has exophthalmos. That's why her eyes are bulging out. She's very thin. And with Graves' disease, when you have too much thyroid hormone, you lose weight even though you're eating a lot of food. You are basically hypermetabolic. And it's because of the excess of thyroid hormone. And these are other examples of endocrine dysfunction due to hyperfunction. You can have pituitary adenomas, and I previously showed you all the hormones that are made in the pituitary gland, all the stimulating hormones like TSH, FSH, LH, and you can have a situation where you're making too much of those hormones. I already showed you what happened when you make too much parathyroid hormone. And you can also have nodules of the thyroid or adrenal gland that can make too much hormone as well. And then there can be malignant hyperfunction, such as with adrenal cancer, and autoimmune, such as with Graves' disease. And then the leading cause of too much cortisol, which is Cushing syndrome, is we give people prednisone and other steroids. And you can also make patients hypoglycemic by giving them insulin or other medications. And there are also inflammatory causes of hyperfunction, such as subacute thyroiditis. And this is the opposite of hormone excess, and it's hormone deficiency. And this patient has hypothyroidism. And as you can see, the prior patient lost a lot of weight, and this patient's gaining weight and has edema, and that's why he has all the swelling in his face. And he just looks kind of sad and slow. And that's also what happens when you have too little thyroid hormone, is it can cause depression and cognitive dysfunction. And this is after treatment. And as you can see, there's a marked difference in his weight and the edema that he previously had, and he looks a little happier too. So there are many causes of endocrine dysfunction that lead to hypo or underactive function. So one of the most common ones is Hashimoto's thyroiditis, which is an autoimmune hypothyroidism. Type 1 diabetes, which is an autoimmune, basically attack on the pancreatic cells, so they stop making insulin. Addison's disease, which is, leads to decrease in cortisol and mineralocorticoid production, or basically all the layers of the adrenal gland, except for the medulla. The medulla functions properly in Addison's. And you can also have polyglandular failure. There are also iatrogenic causes, such as radiation-induced hypopituitarism and surgical hypothyroidism. If you take out someone's thyroid, they're going to be hypothyroid. And there's also infectious and inflammatory etiologies. The leading cause of adrenal insufficiency, not in the US but in the world, is tuberculosis. So you can have infections that also destroy glandular function. You can have hypothalamic sarcoidosis, where you get infiltration of the hypothalamus. And as you saw in the previous slides, you need hypothalamic hormones in order to stimulate pituitary hormones. So that can shut down the entire hypothalamic pituitary axis. You can have enzyme defects such as 21-hydroxylase deficiency, which can lead to hypoadrenalism. And then developmental defects such as Kalman syndrome and Turner syndrome, which can also lead to decreased hormonal function. And then you can have hemorrhage or infarction. And Sheehan syndrome is when a woman infarcts her pituitary after delivering a baby. And it can be an endocrine emergency if she loses all the pituitary function. And you can also have adrenal insufficiency if you have hemorrhage in both of your adrenal glands. And then the last thing is hormone resistance. And this is a case of pseudo-hypoparathyroidism, where there's a resistance to parathyroid hormone. And these patients usually have short stature, and their fourth and fifth digits are much smaller than the rest of their fingers. And then probably the most important hormone resistance is type 2 diabetes, where patients have high insulin levels, but their body is resistant to the insulin, and as a result, their blood glucoses are high. And you can also see leptin resistance when some obese patients are resistant to the effects of leptin. So lastly, I wanted to talk about the clinical relevance, and some of these slides are not in your handout. So this is just what the percentage of obesity was in 1994. And as you can see, this light blue is 10 to 14%. This is 15 to 19%. And this was 20 years ago, so it wasn't that long ago. And as you can see, now it's more dark blue, more, and now there's a different color. In 3 years, all right, I'm just going to test my geography here. I believe I'm going to go with Mississippi, West Virginia. No, this is Kentucky. All right, well, there's some states that are not New York that are between 20 and 24% obese. I grew up here and lived here basically my whole life, so so they're 20 to 24% obese. And as you go along, more states that are not New York that are also between 20 and 24%. And Alaska finally entered the collection, and it's between 20 and 24%. So now you can see, I mean, this is almost, if this were a virus, this would be a pandemic, but it's not. So it's, as you can see, it's growing a lot. Now all of a sudden there's another, and somebody said what state that was, Mississippi, right? So this is 25 to 29%. And as you can see, and now you look in 2005, so only 11 years after we started collecting data, now it's greater than 30%. And this I know for a fact is Louisiana. And this is Mississippi. And Kentucky, I heard what you said, so we'll go with that. Oh, West Virginia, all right. Luckily, this is not a geography class. So, so you have greater than 30%. And as you can see, now when I used to present this up until 2 years ago, because 2 years ago they came out with 2010 data. So in 2009, this I know is the great state of Colorado, is still between 15 and 19%, but not anymore. So I know, it's tragic, it's tragic. So there are no states in our country with less than 20 to 24% obesity anymore. And in about another 20 years, there probably won't be any with less than 30. So this is, endocrinology, if you go into, you'll always have patients. So just to go over some of the other endocrine diseases. So diabetes mellitus, as the obesity epidemic, as people have more and more obesity, they'll also have more and more diabetes. So these were estimates from maybe a couple years ago. The most recent estimates are probably even a little higher. So about 25 million children and adults have diabetes, which is about 8% of the population. And it's estimated to be almost double that in 20 years. And hyperlipidemia, 1 in 4 or 1 in 5 people has high cholesterol. Hypothyroidism, 5 to 10% of women, or 0.5 to 2% of men. Graves' disease is less common. It's about 1 to 3% of women and 0.1% of men. And as you can see, thyroid disease tends to be more common in women than men. And with osteoporosis is becoming more and more common as we have a more aging population. About 10 million people have osteoporosis and 34 million have low bone mass or osteopenia. And it's estimated to be about, I think this is billions, $25 billion that'll be related to an osteoporosis-related fracture by the year 2025. And hyperparathyroidism is a little less common. It's 0.1 to 0.5%. And as people wait longer and longer to have children, infertility is present in about 10% of couples. So I was just wondering if anybody knew what endocrine disease, this is JFK. Yes, he did have Addison. So just to go over what Addison's is, which is it's autoimmune destruction of the adrenal cortex. So you lose mineralocorticoid, glucocorticoid, and the androgens, but androgens in men are also made by the testes, so it's not as important. But what you have here in a normal hypothalamic pituitary adrenal axis is the hypothalamus makes CRH, which then stimulates the pituitary to make ACTH, which then stimulates it has some effect on mineralocorticoid, but as you learned in renal, there are many other things that stimulate aldosterone production. So its main effect really are on the other two layers of the adrenal cortex. So it stimulates glucocorticoid or cortisol production. And then the cortisol goes back to the hypothalamus and the pituitary and decreases its own production. But in Addison's, you don't have you don't have normal cortisol production because it's a primary adrenal cortical insufficiency, and you get autoimmune destruction of all the zones of the adrenal cortex. So as a result, even though you have very high levels of ACTH because the pituitary is not getting this negative feedback, you have high levels of ACTH that are trying to stimulate the adrenals, but since the adrenals don't work, there's no cortisol made or very little cortisol made. So what you see biochemically in this situation is low levels of cortisol and high levels of ACTH. And it can also be characterized by hyperpigmentation, and that's one of the reasons that JFK had this great tan, is because you get increased levels of ACTH, and they also are bound to alpha MSH, which stimulates your melanocytes to darken. And so this is George Bush, Barbara Bush, and their dog Millie all had this endocrine condition. It's not contagious. No one knows? Diabetes. That would be a good guess, but no, it's not diabetes. What'd you say? No one? All right, I'll just tell you. What? Yes, Graves' disease. So all three of them had Graves' disease. And I think someone made a funny joke, but they didn't want to share. So with regard to the pathogenesis of Graves' disease, what happens in a normal thyroid axis is the pituitary gland makes TSH, which is thyroid-stimulating hormone, which binds to the TSH receptors on the follicular cells of the thyroid, which then causes release of T3 and T4, which are the active thyroid hormones. What you have in Graves' disease are antibodies, so activated B cells, and these antibodies bind to the TSH receptor and do everything that TSH would do. So they bind to the TSH receptor on the follicular cells in the thyroid and cause excess secretion and production of T3 and T4. Now this T3 and T4, they go back to the pituitary and shut down TSH production. So when you look at a patient with Graves' disease at their blood work, their TSH is almost always suppressed, so their TSH levels are undetectable. But they are independent of TSH regulation because it's these antibodies that are actually binding to the TSH receptor and causing the increased secretion of T3 and T4. And what happens when you have increased production of T3 and T4 is you can get the exophthalmos, which is the bulging of the eyes. You can get a warm and pulsating goiter in your neck. And because there are T3 receptors on the heart, you can also see tachycardia, a fine tremor. And one of the autoimmune manifestations is you can see pretibial myxedema. So I'm too old to know which Jonas brother this is, but it's one of them, and he's holding a glucometer. So, yes, so what kind of diabetes? Yes, I get it. We have Spanish going here. So yes, it is type 1. So when I was in medical school, this is the model they told us for diabetes, that you had a genetic predisposition, you had a precipitating environmental event, and then that led to the overt immunological abnormalities, and you had beta cell dysfunction because of these antibodies, and it led to progression of diabetes, and you lost C-peptide, which, as I showed you in one of the prior slides, C-peptide is cleaved when you make insulin. So if you're not making insulin anymore, it's very hard to have C-peptide. And unlike if you give a patient insulin and try to check their blood work for insulin, you can't really detect whether it's the insulin you just gave them or the insulin that they're making themselves. Whereas C-peptide, you can tell it's their endogenous insulin production because you need, and C-peptide's really only present if you yourself are making insulin. And medical school has gotten a lot more complicated since I was there because this is the modern model, which is that there are interactions between genes and which impart susceptibility and resistance, and there may be environmental triggers, and maybe they act even before these genes, maybe they act after, and you can have variable insulitis and beta cell sensitivity to injury, and all of these things together can lead to diabetes. So it's not as straightforward as we thought, where you come up with the genetic predisposition and then have all these steps. It seems to be not so much stepwise as we thought, and there may be many immune regulators that lead to diabetes. So the reason that endocrine is important is because it's common, it's preventable, and it's very treatable because we have hormones that basically can supplement any missing hormone that a patient may be missing. So usually endocrine diseases are slow but progressive, but endocrine emergencies do occur. And diabetic ketoacidosis is, if you train in basically any residency, you will see diabetic ketoacidosis. And it's one of the diabetes cases. So it's diabetes case 1 in small group, and we spend the entire time talking about just that one case of diabetic ketoacidosis because it's very important to know how to manage this because it can lead to death, especially in the pediatric population. And Addisonian crisis is if you're not making enough cortisol. The normal human body, which has an intact hypothalamic pituitary adrenal axis, doubles or triples its production of cortisol during stress. And if you're not able to do that, then you can't maintain a blood pressure when you become sick. So many times people with Addison's disease don't present when they're doing well. They present when they get sick and their body cannot mount the blood pressure. And thyroid storm, as its name implies, is when you have too much thyroid hormone. And this can also, it used to have 100% mortality, but now that we know how to treat it, patients tend to do well as long as you give them the right medications. And myxedema coma is the opposite. It's when you don't have enough thyroid hormone and you can present comatose. And hypocalcemic seizures, if your calcium is too low, or if your calcium is too high, you can also present with life-threatening complications. And pituitary apoplexy results in the loss of all your pituitary function, which can be deadly mostly because of its effects on cortisol. If you don't make ACTH, you're not going to stimulate cortisol production. So just to summarize, endocrinology is a study of glands, hormones, and hormone action. The main hormones are peptide and protein hormones. They're also steroid hormones and amines. And they work at either a membrane receptor or a nuclear receptor. And the feedback mechanisms, again, this is something that's very important to learn. And negative feedback is the most important feedback mechanisms and controls most of hormone production. And endocrine disorders are caused by either an excess of hormone, a deficiency of hormone, or a resistance to a hormone. So thanks for your time. This is my contact information, so feel free to email me throughout the section with any questions or concerns you may have. So I don't know if anybody has any questions. Yes, do you have a question?
So real quick, what is myxedema?
It's basically it's a name that the edema part is you get edema in your pretibial region. So that's basically what the name implies is usually, so myxedema coma, you present with edema and you're comatose, so. Sure. We'll go over, obviously, everything that I've talked about in detail when the lecturers do their specific sections. Anybody have any other questions? All right. Actually, this mic is not working that well. So
Can I
I'm
use
just
this?
using that, yeah.
All right. It's on. I should turn it off.
Yeah, it's on right now.
Is it off now? Okay.
It is off now. But this one records everything.
I can put my slides on in between, right?
Yeah.
Okay. I didn't take that. It looks different. Oh, yeah, the desks look different for some reason. I don't know why.
I can't remember if we said I was going to do more things last year.
I don't know. You did from the year before to last year.
I did.
And then I think you decided it was okay.
But if you want,
because you added a little more on like growth hormone stuff, but
And I added some
decided
more.
it was fine.
but you think I should more?
I'll let you know. Yeah, I know. It's like the year comes by so fast.
I
It's
know.
like, oh.
I added some more reproduction to where they changed some of those. But it okay, well, you'll see. Roger the logo's got a really nice recent review where he has everything marked up.
This is like for the ACGME. That's great. So you can actually like do something and make changes on your own. You've been doing all the tallying for the fellows, like with the hospital you meet and all that kind of stuff. All the what? So you can go to like the meetings for like program directors, endocrine program directors and stuff like that. Okay. Okay. Yeah, I think that sounds great. You know, because like I've been on that ESAP committee, the endocrine, and there's like a exam and training and stuff. I mean, I don't know like what falls under the training grant. It's really research. This is all the clinical. So ESAP should be part of that. So that there's like a training exams that comes with ESAP. You have to pay for it, but all the other programs the fellows are doing it. You know, like Susan Mandel's there, all these other people who are program directors for other training programs, and they all do that ESAP exam. So you can use that as like a training tool. Somebody's got to pay for it, but still. Yeah. Yeah.
And also, there's stuff that we have to do, the evaluations and back and forth, and so it's going to have to
Never heard of that.
Right.
Right.
Yeah, should I? And finish quarter up?
Is that on? Okay, good morning. I think we'll get started now. Good morning. Yeah. I'm Sharon Wardlaw from the Division of Endocrinology and in the neuroendocrine unit here, and I am going to give the first of two lectures on the pituitary. I'm going to go over pituitary physiology, and then Dr. Pamela Freda is going to talk about pituitary pathophysiology. And then you're going to have some nice cases demonstrating this in your small sessions later on this week. So just to get started, this is an overview of the hypothalamic pituitary unit. And it shows, you know, how the pituitary is connected to the brain and its relationship to the hypothalamus. And the pituitary is divided into two lobes. We have the anterior lobe and the posterior lobe. And the anterior lobe is the source of many anterior pituitary hormones. And you can see it's connected to the hypothalamus, but not by any direct neural connection. And there are a lot of hypothalamic neurons that make releasing factors
but they're not directly connected to the pituitary. They actually secrete these releasing factors into these vessels, the hypophyseal portal circulation. So this is the pituitary is connected by this stalk, and these portal vessels deliver these releasing factors to the anterior lobe of the pituitary. In contrast, the posterior lobe actually has direct neural connection to the hypothalamus and actually stores peptides in hormones released from these neurons. So in the anterior lobe, we make LH and FSH. These are gonadotropins, luteinizing hormone and follicular stimulating hormone. And they are the same hormones in the male and the female, and they stimulate ovary, testes, and both gonads. So these are called gonadotropins. Prolactin is also released from the anterior pituitary and is responsible for milk production primarily and nursing. Has some other metabolic effects, but this is its primary effect. TSH, thyroid stimulating hormone, stimulates the thyroid. ACTH, adrenocortical stimulating hormone, stimulates the adrenal to make cortisol. And we have growth hormone, which stimulates growth and has a variety of other metabolic effects. So these are the primary anterior pituitary hormones. And from the posterior pituitary, we have vasopressin and oxytocin. And vasopressin is responsible for, it has some vasopressor action and can affect blood pressure, but primarily we think of it as controlling water balance. And I'll show you how it's very critical for that. And then oxytocin, which affects milk letdown and also uterine contraction. So these are the hormones that are coming from the pituitary. And the next slide is just a table of all the anterior pituitary hormones, just for your reference, so that you can see that these hormones come from specific cells of specific cell types. We have the corticotropes, the somatotropes, lactotropes, thyrotropes, and gonadotropes. So these are distinct cells which synthesize these hormones. And there are tissue-specific transcription factors, which are important for expression of these hormones. And it's more if you're a pediatrician and you see hypopituitarism, in children, you often want to think about, could there be abnormalities in any of these transcription factors? For instance, PIT1 has been shown to cause hypopituitarism, and you can see if you have a deficiency of that, you might have deficiencies of somatotrope, lactotropes, and thyrotropes. The hormones which are made from these cells are polypeptides. These are protein hormones. Pro-opium-melanocortin, the precursor of ACTH, growth hormone, prolactin, TSH, and FSH, and LH. And TSH, FSH, and LH are actually glycoproteins, and they have an alpha and a beta subunit. And TSH, FSH, and LH actually share a common alpha subunit, and they have specific beta subunits which give them their specific biologic functions. And this just shows you, we know the chromosome locus, where these are located. It gives you the relative size of the precursors, and shows you here what stimulates and inhibits the hormone release. And we're going to go over this in detail for each hormone, but this is just a summary of the different stimulators and inhibitors of the hormones. And this shows you the target organ, with the adrenal for the corticotropes. Somatotrope has liver and bone and other tissues. Lactotrope, breast. Thyrotrope, the thyroid, and the gonadotrope, the ovaries and the testes. And this shows you what it is that the hormone does. It stimulates the adrenal to make steroids. Here, growth hormone stimulates IGF production, also has some independent growth hormone-mediated effects, which we'll talk about. Prolactin causes milk production. TSH causes T4, thyroid hormone synthesis and secretion. And the gonadotropes cause sex steroid production, but also follicular growth and germ cell maturation. So this is just a little summary table of all the anterior pituitary hormones. And this slide puts it in a more dynamic context. So this shows you that, yes, we have all these anterior pituitary hormones, but they are stimulated by specific hypothalamic releasing and inhibited factors. And you can see CRH is stimulating ACTH release. CRH is corticotropin releasing hormone. TRH is thyrotropin releasing hormone that stimulates TSH. GnRH, gonadotropin releasing hormone, stimulates LH and FSH. Growth hormone releasing hormone, GHRH, stimulates growth hormone release. And you can see I have these arrows are all green for stimulatory, but prolactin is primarily under tonic inhibition by dopamine. And these are the predominant releasing and inhibitory factors. And as you see, when we get into the other axes, there'll be some inhibitory factors as well for some of these other hormones. But if you were to like totally cut the pituitary stalk, so you just cut it here, you would see that ACTH would go down, TSH would go down, gonadotropins would go down, growth hormone would go down, but prolactin would go up. So I think that shows you that these are really the predominant controllers of the release of these pituitary hormones. And once the releasing factor is released, it stimulates the pituitary to make the peptide hormone. It will then stimulate the target gland. And you can just see for every single target gland here, whether it's the adrenal releasing cortisol, the thyroid releasing T4 and T3, testes making testosterone, ovaries making estradiol, there's all feedback effects. So all of these hormones feed back at the level of the pituitary in the hypothalamus to try to regulate their own secretion. And it's an important concept which is going to come up over and over again as we go through all of the endocrine sessions in axes. And this is a slide just giving you the anatomy of the paracellar region. And this is going to be very important for when Dr. Freda tells you about pituitary pathology and pituitary tumors. You need to know where the pituitary is so that you can figure out what's going to be affected by any kind of mass lesion in this area. And here is the pituitary sitting in the sella turcica. And you can see right on top of the pituitary, not too far away, is the optic chiasm. And so clearly we worry about visual problems if there's any abnormality in the pituitary. And on the sides of the pituitary here laterally, you can see is the cavernous sinus. That's where the carotid artery runs. But you also see a number of cranial nerves here. And that's also very important because if you have a mass lesion here, it can invade the cavernous sinus, and you can end up getting problems with these cranial nerves, which Dr. Freda will tell you more about. But just from the anatomy, you can tell what types of things you might expect if there is a mass lesion here. And actually, the sphenoid sinus here shows you this is a nice approach to the pituitary for surgery, which you'll hear more about. And this is just what I showed you, but looking at an MRI. So this is an MRI of the pituitary. Here is a nice normal-looking pituitary gland, pituitary stalk, optic chiasm, and here's the cavernous sinus on either side. And here it's all labeled. So if you have a mass lesion here, you could affect the optic chiasm, or it could invade into the cavernous sinus. And this is just a demonstration of what happens if you do have a mass lesion in the pituitary. So here's a tumor here. And it's affecting the optic chiasm and affecting these nasal fibers are being compressed by the tumor, but they cross here. So you actually get bitemporal field defects. So this is the classic abnormality that you'll see with any kind of mass in the pituitary, bitemporal visual field defects. I mean, obviously, if it's bad enough, you could actually go blind, and so that could be a consequence of a lesion in this area. But we always look for this in patients who we suspect might have pituitary disease. And as I mentioned, this anatomy gives you an approach to how you would actually get out a pituitary tumor. Here is the pituitary. Here's the sphenoid sinus. And you can see the surgeon can go in with a microscope, and he can actually remove tumors transnasally without even doing a craniotomy. So this is the way surgeons can approach the pituitary. So that's just a general overview to orient you to where the pituitary is. And let's now get into the different pituitary target organ interactions, and then we'll go through each one individually. So, but just as a summary, this pretty much summarizes, and you can fill in different hormones, as we go through this, but you have the hypothalamus releasing and inhibitory factors, causing the pituitary to release a trophic hormone, which stimulates the target organ to make a hormone, which then can feed back at the level of the pituitary and the hypothalamus to control its own secretion. And as we talk about pituitary disease or any endocrine disease, whether it's thyroid, whether it's adrenal, whether it's ovary or testes, and somebody comes in with an abnormality, somebody has a low cortisol, low thyroid hormone, low estrogen level, that's what the target organ is making, and we have to ask ourselves, is this a primary problem or a secondary problem? So we talk about primary, end organ disease or secondary disease. And if this is a primary problem, the person had their thyroid removed or their adrenal is removed, there's going to be no negative feedback. So what is the pituitary and hypothalamus going to do? They're going to be revved up they don't know you took out their thyroid, so it's trying to make it work harder to get secretion out of that end organ. So the pituitary hormone levels are going to go up. So you're going to have elevated levels of all pituitary hormones if the target organ is removed. If you have a primary pituitary problem or hypothalamic problem, then the pituitary hormone is going to be low. So we call this secondary, endocrine deficiency. And if you remember this concept, I guarantee there's going to be a couple of questions on your exam that are going to relate to whether it's a primary end organ problem or secondary hypothalamic or pituitary problem. And this is going to be this concept goes through almost every lecture you'll be getting. And just as before we get into the individual axes, just to say, in general, energy balance affects neuroendocrine regulation as well as being sick and stress. But just to concentrate first on energy balance, if a person is starving or has lost a lot of weight, they will have suppression of the hypothalamic pituitary gonadal axis, suppression of the hypothalamic pituitary thyroid axis, and stimulation of the hypothalamic pituitary adrenal axis. And we know some of the underpinnings for this. The fat cells make a hormone called leptin, which you're going to hear about, which tells the brain you have enough energy stores, and engages a whole hypothalamic circuit of orexigenic and anorexigenic peptides, but also engages the neuroendocrine system. So that if we know if you take a mouse and you starve them, and their leptin will go down, but if you give them leptin back, you can correct all of these deficiencies: the gonadal axis, thyroid axis, and adrenal axis. So clearly, that fat hormone leptin is very important in regulating this system. And not only fasting, but severe stress, a chronic illness will do the same thing. You'll end up getting suppression of the gonadal axis and thyroid axis. And in fact, many of our consults as endocrinologists we're called to the ICU, and we have sick people, and you have to decide, do they have real thyroid problems or gonadal problems, or is this just secondary to their chronic illness? So this is also a common theme for most of the axes. And one thing to remember is when we're actually seeing people is this is an example of growth hormone secretion, but this is true for all of the pituitary hormones. They're secreted in a pulsatile manner, and some with a very distinct pattern. And here you can see growth hormone has a lot of peaks at night during sleep, and here's the secretion during the day. And if you were to draw blood on this patient, maybe here, or even if you got them at night when they're asleep, you'd have a, you might just get a snapshot, which might not be to, not really capture what's going on with that patient. And because of the fact that these hormones are pulsatile and any one blood draw is kind of random, we usually do things like either stimulate or suppress so that we can better understand, what the function of that, axis is in that individual patient. And I'm going on now into the growth hormone axis. This is growth hormone IGF, insulin growth hormone factor 1 axis. This is the hypothalamus, this is the pituitary, and this is the hypothalamus. And you can see, as I told you before, growth hormone releasing factor is a primary stimulatory factor of growth hormone release. But there's also another factor, somatostatin, which actually inhibits growth hormone release. This is the primary one because if you did a stock section, pituitary growth hormone would become low. But somatostatin does play a role in fine-tuning the balance of growth hormone secretion. And so growth hormone is released from the pituitary, and it has its own effects on the growth hormone receptor. It can work via the growth hormone receptor. It's a class 1 type 1 cytokine receptor, signals by the JAK-STAT pathway and can have its own effects, but also stimulates IGF-1 synthesis in the liver, and IGF-1 mediates many of the effects of growth hormone, especially on growth. So when we think of growth hormone secretion, we usually think of IGF-1 as an indicator of growth hormone secretion because this is the major factor that mediates many of the effects of growth hormone. And this is showing you the stomach here. The stomach makes a hormone called ghrelin, which also stimulates growth in the growth hormone axis. It's a hunger hormone. It's involved in energy balance, but stimulates growth hormone release. And this is an overview of the physiologic regulators of growth hormone secretion. And green shows you the stimulatory effects, and the inhibitory effects are in red. And if I showed you get a lot of pulses when you're asleep, so sleep is associated with increased growth hormone release. Exercise will increase it as well, and it's something the pediatricians often, sometimes if they're trying to decide if a child has growth hormone deficiency, rather than do fancy stimulation tests, if they run them up and down the stairs and the growth hormone goes up, they may not need to do that. So exercise stimulates growth hormone. Stress also will stimulate growth hormone release, and amino acids in a protein-rich diet. And we sometimes you'll hear an amino acid L-arginine as a growth hormone stimulator. Fasting can also stimulate growth hormone release. So all these things can stimulate. The negative regulator of growth hormone release is glucose. And you'll hear that's a way that if you really want to see is growth hormone too high, is this level not normal, you can give a person a glucose load and see if they suppress normally, because normally glucose should suppress growth hormone release. You can see it's mediated here through somatostatin. And here you see growth hormone releasing factors stimulating, somatostatin inhibiting growth hormone release. And you can see that IGF-1 can feed back and turn off the effects of GHRH. But both growth hormone and IGF-1 can have metabolic and growth-promoting effects. And I'll show you that some of them can be split up. But primarily when we're evaluating people for growth hormone deficiency, especially as adults, we usually use IGF-1 as a surrogate for growth hormone release. But growth hormone does have its own effects. And this is a slide showing the effects of growth hormone and how this works. So we have growth hormone coming out of the pituitary, and it gets to the liver, and it stimulates IGF-1. And the whole IGF-1 story is complicated because IGF-1 binds to a binding protein, IGF-1BP3, and then here's another acid-label subunit which complexes with this. And you don't need to know all these details, but just to know that IGF-1 action is mediated via this complex. And this mediates many of the effects on growth. You also get effects of growth hormone on metabolism, but this shows you all of the way IGF-1 mediates long bone growth. And if you can measure some of these things, but I would say if you just remember that we can measure growth hormone and IGF-1, those are the primary things that we do. Sometimes we can measure this binding protein if we're trying to understand what's going on with the patient. So that's the effects of growth hormone, and these are the effects of IGF-1 and growth hormone, and here's showing effects on the bone. So it affects bone and cartilage metabolism. So you have increased osteoclast differentiation and activity. You get increased osteoblastic activity as well, and increase in bone mass. And you'll get linear growth, you get epiphyseal growth, and increased linear growth. And there is also an anabolic effect where you increase nitrogen retention, increase amino acid uptake, increase protein synthesis, increase organ size, and you also increase energy expenditure as well. So these are all effects of growth hormone on anabolic effects and on linear growth. How about metabolic effects? And this is where you can distinct metabolic effects of growth hormone that are independent of IGF-1. And so these are directly mediated by growth hormone. So in the liver, growth hormone can antagonize insulin-induced suppression of hepatic glucose output. In the muscle, growth hormone can inhibit insulin-stimulated glucose transport. And in adipose tissue, it can cause lipolysis, which releases free fatty acids. And this can impair peripheral glucose utilization. So these things, these direct effects of growth hormone would tend to make your glucose tolerance worse. So that, however, IGF-1 actually is an insulin-like growth factor, right? So IGF-1 can actually cause hypoglycemia actually, and sometimes in kids with hypopituitarism. Without growth hormone, you can see effects on glucose metabolism. And in the liver, there's suppression of hepatic glucose production. And the muscle, it stimulates glucose transport. Here it shows a potent glucose lowering effect and likely has an important role in maintaining normal glucose homeostasis. So here we have this paradox where we have growth hormone making your glucose tolerance worse, IGF-1 making it better, and clinically, when you see a patient with an adult patient with too much growth hormone with acromegaly, which you'll hear about from Dr. Freda, they usually have abnormal glucose tolerance. So there, the high growth hormone seems to trump the effects of the IGF-1. So, but you can see that it's complicated. And and as I said, if you have a child who's growth hormone deficient, I mean, they actually may be hypoglycemic because they don't have the growth hormone. And if you actually give IGF-1 to people to cause to try to cause a growth effect, you can sometimes cause hypoglycemia. So it's a balance, and so when you think of this axis, I would to put it all together, there's metabolic effects, and I think of growth hormone as directly mediating most of the metabolic effects that we see, rather than IGF-1, although it does play a role. And this gives you an overview of how growth hormone and IGF-1 are secreted across the lifespan. And this is important because you can see out here in adults, everything is much lower than it is in kids. And if you're trying to decide whether something is normal, you have to know the age-appropriate normals for these hormones. And this shows growth hormone secretion and IGF levels. going up. This is pubertal, prepubertal adult, and here is out here. And actually, when you order an IGF-1 level on patients, you have age-appropriate normals, and you have to interpret the level within age-appropriate normals. And you can see what's normal for an adolescent can be quite abnormal for an adult. So growth hormone secretion declines with age, and the decreases in both amplitude of the growth hormone bursts, and the serum IGF-1 follows that as well. So that is growth hormone, and I mean, Dr. Freda will talk about this in growth hormone, deficiency and who to treat, and I think we'll also bring this up in the small sessions as well. So, and next, let's move on to prolactin secretion. So prolactin is also made in the pituitary. It's controlled by the hypothalamus, but here it's controlled primarily by a negative factor, dopamine, which is released into the hypophyseal portal blood and inhibits growth hormone release. Actually, TRH, thyroid hormone releasing factor, this may seem random to you, but it's true. I mean, TRH, which stimulates TSH, also stimulates prolactin release. And it's not a primary modulator of this, but sometimes in patients who are hypothyroid and have high TRH may have high prolactin. So that's why it's it's important to think of this clinically. And prolactin works at the breast to induce milk formation. It's induced by the suckling stimulants itself to cause milk production. So this is I'm going to give you the simple version here, and I think if you just remember this, prolactin, dopamine, having this effect on milk, and you can remember TRH. So if you remember this, I'll be happy. This is the more complex version, and the reason I'm showing you this is like Malcolm Lowe's prolactin regulation from the latest Williams textbook of endocrinology, and just to show you how many things can affect prolactin release. Stress, catecholamines, a variety of neuropeptides can affect prolactin release. And this is important to remember if you get a prolactin level which is elevated in a patient. If the patient was stressed, if they're taking a variety of drugs which can interfere with dopamine or a variety of other neurotransmitters, you might get an elevated prolactin level. So you have to really think of this carefully when evaluating a patient with high prolactin. And estrogen itself can directly stimulate the pituitary. So during pregnancy, estrogen can stimulate prolactin release, and this is entirely normal. So during pregnancy, you can have an elevated prolactin, which is perfectly fine, and that's actually important in causing milk production. And this shows you that suckling itself via a neural circuit can directly stimulate prolactin release, so that when a mom suckles a baby, there's a direct physical stimulus that actually causes prolactin release. And if you were to measure prolactin levels during suckling, you'll see that they go up. The take-home message would be the suckling effects, estrogen effects here, and on the next slide, I'm going to parse some of this out. This is the regulation of prolactin secretion, normal regulation. So stimulated by sleep. Actually, sleep gives you mild elevations of prolactin. So if you draw somebody's prolactin early in the morning while they're still asleep, you might get a mild elevation. So something to think about if you're drawing prolactins on a patient in the hospital. Food can give a mild stimulation. Stress can stimulate prolactin release. So if somebody is very stressed with a blood draw or very elevated, they may actually even acutely get an elevation of prolactin. Pregnancy via the estrogen stimulus, like we said, the estrogen stimulates the lactotropes directly. In fact, the pituitary even increases in size during pregnancy because of this estrogen effect, and prolactin will go up. The nursing will do it via the breast stimulation, And sometimes, if patients have galactorrhea and they keep looking for the milk and it's bothering them, breast stimulation itself can actually cause prolactin release. So these are the normal factors that stimulate prolactin release. Prolactin is suppressed by dopamine. It's under tonic dopamine inhibition. So anything that blocks dopamine or prevents dopamine from getting to the pituitary could cause a high prolactin. So a problem here that doesn't let dopamine get down there, or if you give a patient a drug which blocks dopamine effects, you'll get high prolactin. And a prolactin itself can suppress LH and FSH. And here's a normal patient where you have nice normal prolactin pulses and LH and FSH pulses. If the patient becomes hyperprolactinemic, you will suppress FSH and LH. And this happens normally during nursing. The high prolactin suppresses gonadotropins and can give them amenorrhea, but this can happen pathologically as well. And we know that the mechanism for that happening is that prolactin actually affects gonadotropin-releasing hormone secretion. It suppresses secretion. So prolactin works at the level of the hypothalamus to cause decrease FSH and LH secretion. Okay, so that's prolactin. And Dr. Fred is going to tell you a lot more about the pathophysiology of that. Thyroid-stimulating hormone, TSH, is what stimulates thyroid hormone release, and it's made in the hypothalamus, gets secreted into the portal circulation, and causes the anterior pituitary to make TSH, which acts on the thyroid to cause the synthesis of T4 and T3, and both of them can feed back to suppress TSH release. So here you can see TSH is a glycoprotein. It's stimulated by TRH, inhibited by T3 and T4, but also inhibited by somatostatin, dopamine, glucocorticoids, and starvation. So if you have a patient who's getting a dopamine drip in the ICU, they might have a suppressed TSH level, and if they're not eating or when they're on glucocorticoids, all of these things can contribute to inhibiting TSH release. And you're going to hear a lot more about that, as an entire lecture on the thyroid gland. And this is the more, and so just remember, if you remember this, I want you to remember this. This is, again, the more complicated feedback, showing all the feedback loops. Again, showing here, we have the TRH neuron with glucocorticoid receptors, also leptin receptors, and thyroid hormone receptors, and some peptides that mediate energy balance also can affect TRH release. And you can see why we have something called the sick euthyroid syndrome. If you're very sick and these neurons are suppressed, you'll get decreased RH release, decreased TSH release, and low T3 and T4 levels. Okay, now let's move on to the hypothalamic pituitary adrenal axis. And the hormone that is the precursor of ACTH right here. ACTH is only 39 amino acids, but the precursor for this is called pro-opiomelanocortin. So this is a 1000 kilodalton peptide precursor, which in and of itself, if you can't process it, it's not active. It has to be processed, at dibasic cleavage sites by specific enzymes, prohormone convertases, to give you these biologically active peptides. And it's the production of ACTH, a 39 amino acid peptide, which is the primary controller of adrenal steroid synthesis. It works on the melanocortin-2 receptor, MC2, in the adrenal to cause stimulation of cortisol release. And again, the summary of ACTH secretion, a variety of neural inputs and can affect the hypothalamus and stress, many things. But the hypothalamus makes CRH, corticotropin-releasing hormone, which goes into the hypophyseal portal circulation, stimulates the pituitary to make ACTH, which then acts at the melanocortin-2 receptor in the adrenal to stimulate cortisol and other adrenal steroid hormones. And you get a negative feedback of cortisol at the level of the hypothalamus and the pituitary. And importantly, we'll talk about there's a circadian regulation of cortisol secretion. It changes during the day in a predictable way. It's stimulated by CRH, corticotropin-releasing hormone, but actually arginine vasopressin can also stimulate ACTH release. Inflammatory cytokines can stimulate ACTH release, so if somebody's sick or septic, that can be a stimulator. Stress, neurogenic and physiologic stress can stimulate this axis, and cortisol feeds back and inhibits. This demonstrates the diurnal pattern of ACTH and cortisol secretion. So this is a normal person. You can see in the morning they have these nice big pulses of ACTH, and this is the evening. And here you can see cortisol is high in the morning and very, very low. Here's 6 o'clock at night going to midnight. And you can see if you're trying to decide if somebody has too much or too little cortisol, you know, it's very random. If you just try to rely on one single value in the morning, you could totally miss the diagnosis. So we often will measure out here when we're expecting it to be low, and if sometimes we will do suppression and inhibitory tests to try to get around these spikes. And again, the hypothalamic pituitary adrenal axis, this is one is really complicated because there's so many things that can affect cortisol secretion. And things that mediate neurogenic stress, fever, restraint, physiologic stress, starvation. All of these things can feed in and affect the CRH neuron and affect the release of ACTH and then subsequently of cortisol. And this shows you that inflammatory cytokines themselves can directly affect this axis. So sepsis can, and many stresses will affect this axis. So if somebody is very stressed, it may be very difficult to tell if this is a normal response or if they have too much cortisol going on, because this is physiologic for an acutely stressed person. And you may have to wait until the stress goes before you try to figure out if they have too much or not. So this is the axis, and you'll hear again a lot more about stimulation and inhibitory tests. But one of the primary inhibitory tests is going to be giving cortisol to try to feed back and turn this off to see if this can be done normally. And all of the tests that we do, the stimulation and inhibitory tests, are really based on the normal physiology of the axis. So the hypothalamic pituitary gonadal axis is, again, controlled in the hypothalamus by this neuron, GnRH, gonadotropin-releasing hormone, and it releases into the hypophyseal portal blood and stimulates FSH and LH secretion. And these gonadotropins, the same ones, work in the males and females to stimulate ovaries and testes. And there's feedback, which we'll talk in detail when we get to this lecture, of the steroids, at the level of the hypothalamus and pituitary, and also of another factor, inhibin, which is made by germ cells, which feeds back and turns off FSH release. And there is pulsatile release of GnRH. And here it's critical that it be released in a pulsatile manner. And this just shows you, it sometimes in the literature, some people call it LHRH. When you see LHRH or GnRH, they're really referring to the same 10 amino acid peptide. And you can see it's released, in a pulsatile manner into the portal circulation, but it has to be released in a pulsatile manner. And this is an example of when you're giving GnRH in a pulsatile manner, you get nice pulses of LH and FSH. If you then change to a continuous infusion, you totally suppress the release of both gonadotropins, and when you give it back in a pulsatile manner, it comes back again, because you end up downregulating the receptors. So the pulsatile release is critical for normal maintenance of the hypothalamic pituitary gonadal axis. And now just a few words about the posterior pituitary hormones. And all of those axes, which I just went over, you're going to see in a thyroid gonadal lecture, all brought up again. So that was meant just to introduce you to those feedback relationships. So the two posterior pituitary hormones are vasopressin and oxytocin, and they both come from precursors. And ADH, again, they're made in the hypothalamus, in the cell body neurons, which then track down and are stored in the posterior pituitary. And you have ADH, and this is neurophysin is part of the ADH precursor, and that's stored in the posterior lobe of the pituitary. And it works on vasopressin receptors to affect, in the kidney and blood vessels, can affect blood pressure and water balance. And oxytocin, there's oxytocin and another neurophysin, which is part of the oxytocin precursor, works on the breast to cause milk letdown. It also works on the uterus, and it can cause uterine contractions. There are also all sorts of other CNS effects of oxytocin, which you may read about, but that is probably oxytocin made more in the brain and not so much what's released into the peripheral circulation. Effects on behavior, parental behavior. Oxytocin makes people nice. It's a hormone that's studied in lots of behavioral paradigms, but probably that's more the brain oxytocin rather than what's released into the blood. And this is the structure of arginine, vasopressin, and oxytocin. It shows you the structure of the two hormones. They're very similar. And this is DDAVP. This is a vasopressin analog, which is orally active and can be used to treat people who have vasopressin deficiency. And AVP, arginine vasopressin, stimulates, as I said, water balance, affects water balance and blood. And here you can see AVP being released, and it works via the V1 receptor to cause vasoconstriction, so it affects blood pressure, and in the kidney on the V2 receptors, which plays a very critical role in renal water resorption. And hyperosmolarity, hypovolemia, angiotensin II stimulate vasopressin release. And the baroreceptors, natriuretic peptides can feed back and inhibit vasopressin release. So if somebody is hemorrhaging, vasopressin is going to go up because of the blood loss, and you'll stimulate vasopressin release. Vasopressin is exquisitely modulated via osmoregulation. And over here you see a graph with plasma vasopressin in red and thirst in blue. And this is plasma osmolarity. This is normal here, going all the way up as you're getting more, if somebody's getting more and more dehydrated. And you can see as you begin to get dehydrated, or if you fluid restrict somebody, vasopressin goes up. And a pretty low range, too. I mean, this is 6 picograms, 12 picograms per ml. So very low levels of changes in vasopressin when you change osmolarity. And you can see thirst going up in parallel. And over here, you see what happens to urine osmolarity in blue and urine volume in red as you get a change in plasma vasopressin. And you can see at very low levels of plasma vasopressin, 1 to 2 picograms per ml, what happens? Your urine volume is high. This is milliliters per minute per 60 kilograms. So you're putting out a lot of urine. And here is the osmolarity in blue. You're putting out a lot of very dilute urine. And then as vasopressin levels go up, your urine volume really goes down very quickly, and your osmolarity goes up. So you can really concentrate. When you get to vasopressin levels at 4, 5, 6 picograms per ml, you really can maximally concentrate your urine, and the volume goes way, way, way down. So people can easily sit through a lecture without a water bottle. I mean, for an hour, because the important point to make is that I show you the thirst and vasopressin going up in parallel, but if you don't have vasopressin, you can still be thirsty. So it's not, you don't need vasopressin to be thirsty, and people who don't have vasopressin will put out large volumes of urine, but they'll be thirsty, and so they'll drink, and they'll keep up with it unless something prevents them from getting the water. On the other hand, nothing tells you when you're getting too dilute and to stop drinking. So as long as if you drink a ton of water, you will suppress your vasopressin, and you'll put that out. You'll excrete all that free water. But if you have a problem where your vasopressin is high and you can't suppress it, then you'll hang on to the water and get hyponatremic, and there'll be a problem. So this is the last slide showing how vasopressin works in the collecting duct. And it's actually a very neat story. So vasopressin is released from the pituitary, and it binds to the V2 receptor. This is the collecting tubule over here, and this is a blow-up of that. So it binds to the V2 receptor, stimulates cyclic AMP, stimulates PKA, and you phosphorylate this aquaporin channel here. And when this gets phosphorylated, these channels move into the plasma membrane here, and it becomes permeable to water. So when these channels are inserted into the plasma membrane, you can reabsorb the water. If you can't insert them into the membrane, you won't be able to resorb the water, and you may put out 15, 16, up to 20 liters a day if you can't absorb it. So it's these channels which are responsive to vasopressin, which play a critical role in modulating water balance. If you as I said before, if you can't put the channels in and you're putting out a lot of water, as long as you can drink, you'll be okay. But if these channels are inserted here and you keep reabsorbing the water and you continue to drink, you will become then hyponatremic, and there can be a clinical problem. So the peptide, the analog of this, DDAVP, which we use, works very well. People can use it to control water balance, and the urine output will go down and they'll drink normally. However, if they take too much DDAVP and they continue to drink water, they won't be able to excrete that free water load, and they can get into trouble. So this is a really nice system, and AVP is critical for controlling water balance. And it's something that we'll talk about more in some of the cases when they come up. I haven't said much about oxytocin. It's used by the obstetricians to cause uterine contraction, so it's used clinically that way. It can cause milk letdown, although we don't really use it that much to cause milk letdown. And it's being used, and it can be used nasally, and maybe have some effects, some behavioral effects that people are studying. But the major use you'll see of it normally is to cause uterine contractions during labor. So that is I think the end. And if you have any questions about this, that Williams textbook is a really good chapter on the hypothalamus and pituitary. So thank you for your attention.
No, it's this one right here. Yeah.
Okay. Okay, can you hear me? Is that loud enough? No? Okay, can you hear me? Can you hear me? How's that? Okay, great. Okay, so we're going to move on to Pamela Freda, also from endocrinology and the neuroendocrine unit, and give you an overview of pituitary diseases, leading off from Dr. Wardlaw's lecture. And you'll see a lot of the themes also repeat during the specific sessions that you'll have over the course. So let's get this. So Dr. Wardlaw already gave you a great look at the anatomy. This is basically similar to her picture. We don't need to really go over this. And also, she's already gone over all the hypothalamic pituitary axes. So we're going to talk about diseases that affect the pituitary, but also just keep in mind that these can be diseases originating in the hypothalamus. They can be diseases originating in the anterior pituitary and affecting those hormones primarily, either causing a deficiency or an excess. Diseases in the pituitary can affect the posterior pituitary hormones. And then also manifestations of these hypothalamic pituitary diseases will come up primarily because of effects on the target glands. So the clinical manifestations will really be key that you understand, and this will be going on during the whole endocrine session, learning about all the clinical manifestations of target gland effects and how these are changed in different pituitary diseases. So what kind of general diseases can affect the pituitary? Well, first of all, there can be mass lesions, tumors, other kinds of mass lesions, and we'll go over these. They can be primarily coming from the pituitary gland. They can come from the hypothalamic pituitary stalk, which Dr. Wardlaw showed you, the stalk that connects the pituitary to the hypothalamus. Or they can come from the hypothalamus primarily and then downstream affect the pituitary and the target glands. You can have a whole series of genetic abnormalities affecting the pituitary, infiltrative and inflammatory processes, and a series of other causes. And we're going to go through each of these general groups in this lecture. So going into first, what are the tumor and mass lesions affecting the pituitary? So the most common kind of mass that you can have affecting the pituitary are pituitary tumors. And these are benign tumors generally. They can be of sort of two categories, either hormone secreting or non-hormone secreting. And we'll go through those, meaning that they originate in a cell that is producing a particular hormone that is secreted out into the circulation, and we can measure it in the circulation. We call those hormone secreting. And others which are non-hormone secreting. In the blood, we don't see an excess of a particular hormone. Just to make the point, we'll come back to the tumors in much more detail throughout the lecture, that tumors are monoclonal lesions of one particular pituitary cell causing a tumor. This is distinct from what we'd call pituitary hyperplasia, which you'll see in certain conditions. These are not tumors, but where the whole pituitary is enlarged, all the pituitary cells or a large portion of a certain kind of cell are enlarged. For example, in pregnancy, the lactotroph cells can become hyperplastic and enlarged. In primary hypothyroidism, which Dr. Wardlaw told you about, TRH will go up, and that will stimulate the pituitary to cause it to be hyperplastic and enlarged. You can also rarely, and we'll talk about this a little bit on Wednesday when we talk about acromegaly, you can sometimes have secretion of growth hormone releasing hormone from another kind of tumor throughout the body that then feeds back and stimulates the somatotroph cells and makes them hyperplastic. We're not going to talk a lot about these, but just to make it clear that these are not tumors, and these should really be determined by the clinical setting of the patient. So what are some of the kinds of tumors? We'll come back to the pituitary tumors, but there are others that are not originating in a pituitary cell. So we call these non-pituitary lesions. And if you think back, I don't have the anatomy right on here, but Dr. Wardlaw showed you that. There's obviously a lot of structures around the pituitary, the meninges, various different structures, the cranial nerves, everything all around the pituitary. So you can have tumors originating in a lining cell or another kind of cell around the pituitary that can affect the pituitary itself by pressure. So there are a number of other kinds of pituitary lesions. One category are some benign tumors called a craniopharyngioma. You can also, and we'll talk about that more in the next slide, you can also have a tumor of the meningeal cells around the brain called a meningioma that can press on the pituitary and sometimes look like a pituitary tumor. Then you can have various kinds of cysts that can get into the pituitary area. One, the most common, is called a Rathke's cleft cyst, and it's basically a developmental cyst that people have, since birth that fills with fluid and causes a cyst in the pituitary area, so a fluid-filled lesion in the pituitary that sometimes can look like tumors, but more often you can tell they're cystic, and they're not uncommon. Then there's a whole series of malignant tumors that can either be metastatic, coming from another tumor elsewhere in the body, to the pituitary or primary in the pituitary area. There's one called a chordoma, which actually arises in the bone that sits right behind the pituitary, called the clivus, and they're aggressive tumors. And there are a number of tumors that seem to have a propensity for the pituitary, breast cancer, lung cancer, and they seem to most commonly go to the posterior pituitary. So the hormone deficiency, you can imagine from what Dr. Wardlaw just told you, is often a vasopressin deficiency that they come in with, these kinds of tumors, because they affect the posterior pituitary where vasopressin is secreted from. And occasionally we have lymphoma, or other hematologic malignancy that affects the pituitary. Then aside from these other kinds of cysts, you can also have unusual but interesting granulomatous, infectious, or inflammatory things that affect the pituitary. Sarcoidosis can affect the pituitary. Tuberculosis also can affect the pituitary, infiltrate it. Usually involves the meninges around the pituitary. Very rarely an abscess, an infection can affect the pituitary. And then there's an interesting disease called lymphocytic hypophysitis, which is basically a lymphocytic infiltrate that gets into the pituitary. It's more common in women late in pregnancy and can present with a tumor or a mass. What looks like a tumor is really a mass in the pituitary, which is a mass of inflammatory cells within the pituitary. And patients can get pituitary deficiencies, usually in kind of an unusual pattern. So what about hypothalamic diseases? So as we said, you can have a tumor that's coming primarily from the hypothalamus that's going to then downstream affect the pituitary function. So masses in the hypothalamus could be a craniopharyngioma. I'm going to say more about that. And also germ cell tumors. And then we also think about the history of the patient, obviously, because some people may have had radiation to their brain for other kinds of tumors, head and neck tumors also. And traditionally, some of these radiation forms, especially if they were not particularly focused, could affect the pituitary function. So that's a cause of hypopituitarism, having had radiation to your brain or your pituitary. And then, as I said, these other infiltrative diseases, some of them originate also in the hypothalamus and affect the pituitary. Craniopharyngiomas I talk a little bit more about because we see them fairly commonly. They are benign tumors. They originate in rest cells, and basically they're from remnants of this Rathke's pouch, which is a developmental thing where the lining comes together, and developmentally, in some patients develop a tumor from that in between the lining. And they can occur anywhere from the hypothalamus all the way down to the pituitary. They can be solid or solid and cystic. And interestingly, they have sort of a bimodal distribution. There's kind of a peak in children and also in adults. And typically they present with fairly large lesions, with vision loss, signs and symptoms of other pituitary insufficiency. They're generally treated with surgery, sometimes transsphenoidally through the nose, as Dr. Wardlaw showed you the picture of. Otherwise, sometimes they need a craniotomy, cutting through the skull to get to those tumors, depending on how large they are and where they are. And sometimes patients get radiation treatment to the tumor to try to keep it from growing. There are a bunch of other miscellaneous causes of pituitary disease. There's various vascular lesions, such as an aneurysm. If you recall the picture of the anatomy, there are obviously the carotids and blood vessels right around the pituitary. And sometimes aneurysms of those blood vessels can look like a mass in the pituitary. Fortunately, we don't come across this very often now because of MRI. You can really see very clearly what's a blood vessel and what isn't. In the old days, without that, it was a little bit harder to tell what could be a tumor versus an aneurysm. But we usually know after a patient has an MRI if it's an aneurysm or a tumor. Then there's something called pituitary apoplexy, which is basically a hemorrhage into the pituitary or infarction that cuts off the blood supply, but usually we see a hemorrhage into the tumor or into the pituitary. And that usually presents with a very sudden expansion of the pituitary, headache, often vision abnormalities, cranial nerve abnormalities, because something we'll get to, I think, a little later in the talk, pituitary tumors in general are very slow growing, and symptoms are there, but they're not as severe as you'd see with a sudden enlargement or a sudden expansion of the pituitary with bleeding into it. So that's called pituitary apoplexy. And if it's severe and the patient has vision abnormalities and sometimes cranial nerve deficits. You do have to usually operate, the neurosurgeons do, and relieve the pressure of the blood. Some people with a tiny little bit of blood with no symptoms, you can just follow it and watch it get better, usually over time. There's also something called Sheehan syndrome, infarction of the pituitary, which classically occurs peripartum in women and sometimes associated with a lot of blood loss. And that can present with hypopituitarism and sometimes goes undiagnosed many years later. Women show up with, or postpartum with pituitary insufficiency. There is a literature also now on the effects of head trauma on causing hypopituitarism, varying degrees of that, most likely due to some sort of shear stress on the hypothalamic pituitary stalk. There are a series of genetic diseases. We're going to talk about those in a little bit, a couple more slides. And then there's something called empty sella syndrome, which is a little bit of a misnomer. Basically, it is to a certain extent a radiographic diagnosis, and what you see, and next year I should bring a picture of this, is the pituitary. Instead of seeing the nice pituitary in the sella, you see a lot of the CSF, the cerebrospinal fluid, in the pituitary area, kind of pushing down on the normal pituitary, and it's called empty sella. Usually it's not associated with nothing in the pituitary. It's usually associated with the pituitary gland kind of being flattened or pushed down. But it can be associated with hypopituitarism, but not in everybody. But we usually see it on MRI, and then it needs to be worked up for pituitary insufficiency. It's more common in women who've had many children, and it may also occur when there's been a pituitary tumor that has infarcted and shrunk down, and then you're left with a space where the pituitary was. Now, the genetic diseases, you're going to hear more about these from the pediatric group. You don't need to know all of these. I just bring up two of them in the next slide, which are two important genetic causes of pituitary disease. And Dr. Wardlaw did touch on these in her lecture. And one important one is called the PROP1 mutations. And this is really important. It's the most common cause of familial and sporadic congenital pituitary hormone deficiencies. And it's a mutation in a gene encoding a transcription factor necessary for differentiation of all these cell lines, the somatotroph, lactotroph, thyrotroph, and gonadotroph cells. So what you'll see in these patients is deficiencies of all these hormones, prolactin, and usually later on they develop an ACTH deficiency. This is autosomal recessive. The other of the most common of these genetic disorders is called a PRU1F1 mutation. This is autosomal recessive and dominant in different families. It's a mutation in a transcription factor necessary just for the somatotroph, lactotroph, and thyrotroph cells. So these people will see, these patients, growth hormone, TSH, and prolactin deficiencies only. So Dr. Wardlaw talked about this and showed you a picture with the anatomy, and I'll just go over this again. When you have a mass lesion, any kind, some have propensity for different things, which I touched on a little bit, and we'll go over more during the lecture. But any mass lesion within the pituitary area can cause mass lesion symptoms, and signs. And these kind of fall into the general category of visual and neurologic abnormalities. One is headache, and the problem with headache itself is very common, but we commonly see it in pituitary tumors. So it's a little hard to sort out and say a patient with a headache and a pituitary tumor, it's due to that. The one caveat to that is, as I mentioned, the pituitary apoplexy, for example, or another lesion that happens to be unusually rapidly growing will cause headache more likely. So rapid stretching of the meninges and the lining around the pituitary will cause headache. But it's a common manifestation of pituitary tumors. And often it does get better when you take the pituitary tumor out or you shrink it, but it's not a constant finding. The other important deficit are the visual field deficits, which Dr. Wardlaw already showed you picture of, with the classic being the bitemporal hemianopsia. But there can be very subtle defects all the way from a tiny superior defect on one side to to really loss of visual acuity if the lesion is very large and has been let to grow for a very long time. And then there are all the cranial nerve deficits. She showed you the picture with the anatomy with the cranial nerves. And typically, though, pituitary tumors themselves do not present with a cranial nerve deficit. And it might be part of the nature of the lesions. They're very slow growing, and they tend not to be infiltrative. But sudden expansion, such as the apoplexy, or patients with hemorrhage into the tumor can present with these cranial nerve deficits. And also the something important to remember, the non-pituitary and the inflammatory diseases, the other kinds of unusual lesions, the inflammatory things, sarcoid, tuberculosis, those kinds of lesions, or metastatic disease, other things, are more likely to be infiltrative and irritate the cranial nerves. And so we often see these kinds of problems present with the cranial nerve deficits. In particular, the third cranial nerve is the most common, with patients presenting with ptosis, but often the fourth or the sixth in patients presenting with diplopia due to deficit of adduction or abduction from these cranial nerves. So besides the mass lesion deficits, any kind of pituitary lesion is going to have a clinical presentation that in part is going to take on the characteristics of that kind of tumor, if it's a tumor, but also sort of generically, anything that presses on the pituitary itself can cause hypopituitarism. So it could be a pituitary tumor itself, it can be one of these other kinds of lesions, it can be an infiltrative disease. So there are a whole series of signs and symptoms of hypopituitarism, and they really depend on what hormone is being lowered. And not every lesion causes every hormone to be lowered. It can be variable, or it can be all of them. So any one patient with a pituitary disease or a pituitary lesion can have a whole spectrum of pituitary deficits. And we're going to go over some signs and symptoms of hypopituitarism for the different axes. And then patients with a hormone-secreting tumor, which we're also going to go over in a few slides, that particular patient has the characteristics of that kind of hormone-secreting tumor. So whatever kind of cell the tumor started in, whether it's a prolactin cell or a growth hormone tumor, that patient is going to take on those characteristics. So we'll talk about all the different clinical settings for those. The other clinical presentation, which is interesting, is that actually there are many people who have these pituitary lesions, most likely all benign tumors or small cysts, noted really incidentally on a head imaging done for another reason. So actually, if you look at autopsy studies and some actual clinical screening studies, about 1 in 5 people actually has a pituitary lesion and probably doesn't know it. So they're really very common, and it's probably being picked up. I don't know whether they're becoming more common or just being picked up more commonly, because practically everybody who rolls into the emergency room with a bad headache or something is going to get a CAT scan and maybe even an MRI to follow that up. So they're picked up very commonly. So there's something definitely to know about, because it's likely you're going to see a pituitary tumor somewhere along the line. And the best imaging modality, which Dr. Wardlaw also talked to you about, is MRI. You can pick up a lot of pituitary tumors on CAT scan, but we really look for the MRI, and it's really the gold standard. It can look very closely at the pituitary, the surrounding structures. You can see the chiasm, the stalk, the cavernous sinus as well. Sometimes can even see the cranial nerves. And so we do the MRI with and without this gadolinium contrast, which really helps you determine tumor from normal tissue. And I'll show you in the next slide what I mean about that. So here's an MRI, and you saw one already from Dr. Wardlaw. This is something called a coronal image, looking straight at the patient. You can see where the normal pituitary is, the hypothalamic pituitary stalk, and the optic chiasm above it. And here's the sagittal, like slice right down through the middle of the head, showing the anterior pituitary, the posterior, which shows up as this bright spot. It's often bright and because of the high water content in the vasopressin neurons, but it's not necessary to have it in normal people, but we often see it in normal people. And here's the optic chiasm, and here's that stalk. And then when you get a pituitary tumor, what you see is a hypoenhancing, so doesn't pick up the contrast as normally as the normal pituitary. And you see this hypoenhancing lesion also here and here in the pituitary. And this is a small lesion. We call this a microadenoma. It's less than 1 centimeter. And then for pituitary tumors, you can also have a large tumor, greater than or equal to 1 centimeter. And this, again, is the sagittal. And this outline here, this is the tumor. And actually, probably this is the normal pituitary being squashed and lifted and pushed up. And somewhere in here is the optic chiasm. You can see it better on this scan. This is the same patient, but in the coronal image. And here you see probably the normal pituitary squashed to the side, and the optic chiasm is actually draped up over the tumor and compressed. It's thinned, and if you remember back the normal, you can see it's totally disordered. And here's the whole tumor and the carotids on the side. So you can see how a lesion in the pituitary here, and this is not a huge one, this is large, but it's not huge, could really cause the vision abnormalities and hypopituitarism. So let's go over then, and you're going to be learning more about this as you do each system throughout the endocrine course, but just overview of the manifestations of hypopituitarism for the different axes. And so if you have hypopituitarism, as Dr. Wardlaw told you, we call that secondary adrenal insufficiency, because it's not the adrenal glands themselves, it's the pituitary being affected that leads downstream to adrenal insufficiency. So it's ACTH deficiency, which leads to decreased adrenal cortical production. And the symptoms are really of adrenal insufficiency. Patients have lethargy, weakness, poor appetite, nausea, weight loss. They can become dizzy, hypotension and hyponatremia, low sodium, and you're going to hear more about this in your talks, and these are all symptoms of what we see with cortisol deficiency. And the treatment of this is basically replace them with glucocorticoids. There are a lot of different preparations for giving them back glucocorticoids, which is basically the general category for cortisol and cortisol-related hormones. We often use oral hydrocortisone, which is the natural cortisol produced by the body, but there are other things. Prednisone is synthetic steroid. There's cortisone acetate. There's other things you can use to replace the steroids. Then the other hormone, which also you'll learn more about, is thyroid deficiency, so secondary hypothyroidism, which is due to abnormal TSH secretion from the pituitary and then result in thyroid hormone deficiency. And patients have symptoms of thyroid insufficiency, fatigue, dry skin, constipation, bradycardia, slow heart rate. They feel cold all the time. Then we also see something which is very characteristic of hypothyroidism, which is delayed relaxation of the deep tendon reflexes, and a puffiness of the soft tissues is characteristic. And to treat this, we administer oral thyroid hormone replacement, which is a daily medication. Some levothyroxine, L-thyroxine is the typical form given, and I think you'll learn more about this when you talk about thyroid. And then another axis which can go out when you have hypopituitarism is the gonadal axis, so we call that secondary hypogonadism, and there's a spectrum of clinical manifestations depending on how severe, the age of the patient, and obviously the gender. In women, having secondary hypogonadism can give you symptoms of irregular periods all the way to no periods, amenorrhea. Some women with hypogonadism, secondary hypogonadism, and infertility. They can have, obviously, estrogen deficiency because the ovaries are not receiving the normal signals, so patients don't produce. They're not receiving the LH and FSH, the estrogen from the ovaries. And estrogen deficiency can lead to osteopenia, low bone mass, like a woman with true menopause leading to osteopenia or osteoporosis, but in this case, in a woman before the menopause, it would be due to the hypopituitarism and estrogen deficiency. And then in men, you also have the whole spectrum of disorders, from sexual dysfunction, decreased libido, gynecomastia, which is enlargement of the breasts, can occur in men, basically due to development of an imbalance between the androgens and estrogens in the men. And I think you'll be talking about that more in the course. Men can also have infertility because they're going to have low gonadotropins, therefore low testosterone, low sperm count, impairment of fertility on those bases. And then also men can also get osteopenia and low bone mass from low testosterone and low estrogen, which results from hypopituitarism. The way we treat hypopituitarism and the secondary hypogonadism is replacing with testosterone in men. And traditionally, we gave testosterone as an injection, depending on every 1 week, 2 weeks, 3 weeks. Now we have all these topical gels. I'm sure you've seen it on television. Lots of ads for this. And those testosterone, preparations are actually used properly to treat for hypogonadism in men. So that's what we use all those under the skin, under the arm or on the skin, daily testosterone supplements. And women, really, how you treat it depends on their age. So young women, postmenopausal women, you will make a decision to treat, their gonadal steroids. It's a different situation, but in premenopausal women, you're going to look to whether you want to restore fertility or just give them back the estrogen and progesterone. And you're going to have other lectures about this, but you may need to do fertility treatments to stimulate the ovary with HCG or gonadotropins in women who are looking for fertility versus women who are not looking for fertility. You may just replace the hormones, the estrogen and progesterone. So it depends on the clinical setting. And then for growth hormone deficiency, growth hormone is probably the most controversial hormone of the pituitary hormones. Who do you replace and when and why? Because it's not life-threatening, really, growth hormone deficiency. And you need to, in adults at least, let's leave the kids aside, but talking about adults, the clinical manifestations are really mild. The growth hormone deficiency, if you think back to what Dr. Wardlaw had on her slide about all the metabolic effects of growth hormone burns fat, so it's lipolytic. So actually what patients with growth hormone deficiency have is a mild increase in fat mass, and it's mostly essential fat mass. And they also have decreased lean body mass because growth hormone is anabolic and puts on muscle to a certain extent. So patients with deficiency have slightly low lean body mass. Their cholesterol and cardiovascular risk markers are also a little bit higher, and they can have mildly decreased bone density. So it's controversial because these clinical manifestations in adults are not life-threatening, and they can be subtle in a patient, but in the right patient who clearly has clear growth hormone deficiency. And I just will say that usually growth hormone deficiency occurs with all the other hormones. So usually you've identified a patient who has pan-hypopituitarism who also has growth hormone deficiency, at least in adults. And so you look at all these clinical manifestations and make a judgment about whether the patient could benefit or not from growth hormone. But this is very expensive. It is an injection every day. And so it's a big commitment for the patient and otherwise to treating. So it has to be considered carefully. This is separate from what you're going to hear about in children. Obviously, growth hormone is essential for growth, and in children, it's a different story. There's not so much question about treating, and you'll hear about that, I think, on Wednesday. And as I said, you need to give daily injections. And because you're injecting growth hormone, you're going to get some large ups and downs of growth hormone, and what you follow when you treat them is the IGF-1 level, because that gives you kind of an integrated measure of how much growth hormone they're getting. Growth hormone is not the only factor that stimulates IGF-1, but in this setting, it should be on the lowish side for growth hormone deficiency, and you'll treat to normalize that level to a reasonable level and make the patient overall feel better. And then Dr. Wardlaw talked a lot about vasopressin, but just to talk again a little bit about what happens when you have deficiency of vasopressin. So actually, pituitary tumors before surgery or before any manipulation of the pituitary area don't commonly cause vasopressin
vasopressin deficiency, but we see it often after surgery for the pituitary tumors. We'll get into that, the different kinds of tumors in the surgery. But basically manipulation of that area, and also the infiltrative and inflammatory diseases, and the metastases tend to go to the posterior pituitary. So we see it more commonly in that setting. But basically what we see clinically in the patients, and this, for example, somebody who's had pituitary surgery, we might see the next day. They're in the hospital, they had the pituitary tumor taken out, and there was a lot of manipulation in the area. So that hypothalamic pituitary stalk was very disrupted, and we often see some vasopressin deficiency, usually transient. But what you'll see is that they urinate large volumes of urine, liters and liters sometimes if it's very severe. They become very thirsty to compensate. What we do to test, we measure the urine volume, the specific gravity, the osmolarity. Frequently, we measure the serum sodium, which if the patient is drinking and can keep up, should not get elevated. If it does get elevated, then we haven't corrected the water deficit properly. In a patient where it's not obvious that they have it, say someone coming in with one of these unusual problems, we don't know them to have a pituitary lesion, you might need to diagnose a vasopressin deficiency by actually dehydrating the patient intentionally. So bringing them in a controlled setting and keeping them from drinking over a period of time, and following their urine output over time, and looking to see what happens to the concentration of the urine. And then if you give them DDAVP, the synthetic vasopressin, you'll see that they'll be able to concentrate the urine over time. And so that's a kind of complicated test, but we have to do that sometimes to diagnose this. And what we see on laboratory testing for vasopressin deficiency is if they haven't had access to water, such as if you had a patient who had vasopressin deficiency and you brought them into the hospital, for example, and they didn't have access to water, they could get dehydrated very quickly over the course of a day. And so you'll see the serum sodium level get up if they're unable to drink. And you'll see this very dilute urine despite them having a high sodium and being dehydrated. And we treat it with the DDAVP, long-acting analog of vasopressin, or some pills. This nasal spray is more potent and works more quickly, so people with severe deficiency usually use that, but we also have some pills. So what about pituitary tumors? So pituitary tumors are, as I said, the most common cause of lesions in the pituitary. And they are benign tumors originating in one particular pituitary cell. They're monoclonal in origin, that is, they originate in one cell. And the type of tumor they are depends on what cell they originate in. The lactotroph cells, those produce the prolactin-secreting tumors. So tumor originating in the lactotroph cell will be making prolactin and a high level of prolactin in the blood, and so on down this list, which you can read, of the different kinds of tumors and what cell they originate in. Pituitary tumors are more common in adults. They do occasionally occur in children. And interestingly, they are overall more equally common in men and women, although there are some kinds, and particularly the ACTH-producing ones, that are more common in women. Generally, they're sporadic, just come up due to mutations within the pituitary cell itself. We don't understand all the mechanisms, but there are some familial syndromes that are associated with somatic mutations that cause pituitary tumors. There's the multiple endocrine neoplasia type 1 syndrome that's associated with parathyroid tumors, pituitary, and other endocrine tumors. And that's an autosomal dominant mutation in the MEN1 tumor suppressor gene. There also are some relatively newly found familial pituitary tumor syndromes with just pituitary tumors associated with certain other kinds of mutations. And as I mentioned, pituitary tumors are probably as common as 1 in 5 people in the population. So what about the different types of pituitary tumors? There's a group of tumors we call clinically non-functioning tumors. And what I mean by that is they are not dead or sitting there not functioning, these tumors. But basically they are producing, not producing a high level of a hormone that we can at least detect in the blood. And they are not producing a clinical syndrome related to the high level of hormone. And that will make sense to you as we go through all the different clinical syndromes. But even though they're clinically non-functioning, most are actually coming from the gonadotroph cell line. So that there's a small percentage that do make some of the gonadotrophs into the blood, or not into the blood, but actually if you stain the tumor, you'll see the gonadotroph cells, but they don't actually cause a clinical syndrome. So they're still non-functioning. And because they don't have a clinical syndrome associated with them due to all the hormone excess, they often present when they're large. So if you see a very big tumor and the patient really doesn't have signs of Cushing's or acromegaly, which we'll go into, they're often a non-functioning tumor. They present with the hypopituitarism due to compression and vision abnormalities. The treatment of the symptomatic tumors, of these tumors, when they're large enough to cause symptoms, is the surgery to remove them. And they do sometimes come back, though. Not uncommonly, we need more surgery or radiation to treat them. There are, within the non-functioning tumors, there are rarely some gonadotrope-secreting tumors that do produce a clinical syndrome. They're not common. They could be LH-secreting, which in some cases have been reported to raise the testosterone and cause precocious puberty. That's early puberty in males. And rarely they could be FSH-secreting tumors and produce ovarian stimulation in women, but those are very uncommon. So I'm going to talk a lot more about this type of tumor on Wednesday, but just to give you an introduction to it today, a growth hormone-producing tumor originating in a growth hormone cell is a disease we call acromegaly. When you get it in children, it causes gigantism. In adults, it's acromegaly. And basically, it is characterized by high levels of growth hormone and high levels of IGF-1. Dr. Wardlaw went over this axis for where these all come from. And we'll go over on Wednesday, when I talk about abnormal growth, a lot of things about acromegaly. And then you'll also learn about this in your adrenal section. There's a tumor of the corticotroph cells. It causes a disease called Cushing's disease. And basically, the high level of ACTH produced by the corticotroph tumor leads to a high level of cortisol in the blood. And Dr. Wardlaw went over this axis, and you'll learn more in your adrenal section about Cushing's, differentiating a pituitary source of Cushing's from other kinds of tumors and throughout the body that can produce ACTH, something called ectopic ACTH. But Cushing's disease is the name for the ACTH-producing pituitary tumor that causes Cushing's disease. I just have, I guess, one or two slides about the manifestations of Cushing's, but you will learn more about it. Basically, it's characterized by a whole systemic series of clinical manifestations due to high levels of cortisol throughout the blood. Patients develop obesity centrally around the middle, fullness of the face, thin skin, bruising, proximal muscle weakness, so that when they are sitting in a chair and they try to get up, they can be weak. They don't use their arms. They get glucose intolerance because cortisol antagonizes insulin effects and can cause diabetes, high blood pressure due to the high cortisol. And as I mentioned to you, it has a female predominance, and most cases of Cushing's disease are diagnosed in women between 20 and 40 years of age. We diagnose this traditionally by, and Dr. Wardlaw, she mentioned to you about the pulsatile nature of ACTH, but also the diurnal rhythm of cortisol. So just measuring a random cortisol sometime during the day is not going to be very helpful. But what we use is an integrated measure of cortisol secretion. So that happens to be the 24-hour urine-free cortisol. And I think you'll learn more about this in your adrenal section, but we diagnose it with a high level of urine-free cortisol. And what you'd see from a pituitary tumor causing Cushing's is a normal or elevated ACTH. And I think you'll go over this in your section, whereas if the adrenal was the source of Cushing's, the ACTH would be low. And you may need a number of localization studies, something called petrosal sinus sampling, which you'll learn about, to see if the pituitary really is the cause of ACTH, because it can be coming from other kinds of tumors in the body occasionally. What we do for Cushing's is remove the pituitary tumor. The neurosurgeons do. They go with the transsphenoidal, which you were showed a picture of, and try to find the tumor. They're usually small because these patients have a lot of clinical manifestations that are very severe. Unlike the non-secreting tumors, the Cushing's have all kinds of clinical problems, the obesity, diabetes, et cetera, thin skin, which makes them picked up much earlier when they're tiny. So they can be hard to find, and it can be challenging clinically to evaluate them. And occasionally, if they're not treated successfully with surgery, we do radiation or even take out the adrenal glands, bilateral adrenalectomy, if they cannot be cured otherwise. So what about another kind of pituitary tumor, the thyrotropin releasing hormone, or TSH secreting tumors? They originate in the TSH secreting cells. These are rare, but they often present with a pituitary tumor that's usually large, and they have symptoms of hyperthyroidism. So the TSH is elevated, drives the thyroid itself to raise the thyroxine, and so you get symptoms of hyperthyroidism. And those include tremor, shaking, sweating, very smooth skin. Patients have like a stare to their eyes. They get palpitations and weight loss. And they also have a goiter, which is an enlargement of the thyroid gland, because the thyroid is being stimulated by the TSH. And what you see on blood levels is an elevated level of thyroid. But you also see, and you'll learn about this in the thyroid section, as Dr. Wardlaw was talking about, looking at the pattern of the pituitary versus the end organ hormones is very important for trying to figure out where these different problems are coming from. So in a TSH secreting tumor, the TSH is what's driving the hyperthyroidism. So it's actually normal or elevated, and the thyroxine is elevated. This can be a challenging thing to try to sort out, even for the endocrinologists in some patients. And what we treat these with is transsphenoidal surgery to remove the tumor. They can also be treated with radiation or somatostatin analog, because as Dr. Wardlaw told you, somatostatin actually suppresses TSH, so they can work for some of these tumors in treating the tumor. So just to give you an idea of what it looks like in some of these tumors, look at another MRI. This is an MRI after surgery, a large pituitary tumor here, and this is after surgical removal. And they couldn't get this whole piece around what's basically in the cavernous sinus, but you can get a lot of the large piece, and sometimes they do more invasive surgery and get that piece of pituitary tumor. And you can see if you're left with a hormone-secreting tumor here, you're going to need to do radiation, other types of treatments, maybe more surgery to get that out. So now I just have to quickly go through hyperprolactinemia. And Dr. Wardlaw talked about the physiology of this, but I'm just going to touch on some clinical aspects of hyperprolactinemia, too high prolactin levels. And basically it's diagnosed as a prolactin that's over 20 in men or 25 in women, and that depends on what laboratory you're using, what particular assay you're using to measure it. But it's really a very common endocrine disorder of the hypothalamic pituitary axis, probably the most common. And if you just screen a large group of adults, you'll find it supposedly in 0.4%. And there are really a lot of causes of hyperprolactin, as you saw, but prolactinomas, the little prolactin-producing tumor, is the most common cause. So what are some of these pathologic causes? We're talking here about prolactinomas, but in terms of hypothalamic pituitary disorders, people with acromegaly, growth hormone-secreting tumors, can also have hyperprolactin, or really any other cellular mass. And that makes sense because what Dr. Wardlaw told you is it's the only pituitary hormone that's normally kept low in your blood. And when you have a lesion in your, or normal, when you have a lesion anywhere from the pituitary through to that stalk to the hypothalamus, is going to interfere with the dopamine signals coming down to the pituitary. It's going to raise the prolactin. So that raising of the prolactin can really occur with any of the other pituitary lesions. And when the prolactin's in a range of something up to maybe 150 nanograms per ml, we generally call it what we call a stalky prolactin, or a prolactin that could be due to one of these other causes. But there's a lot of overlap in prolactin levels between tumors and stalk, so it's kind of complicated to sort out. But just get the general concept that other things besides a prolactinoma can raise the prolactin. And then there are a whole bunch of other causes, which Dr. Wardlaw talked to you about, related to the normal physiology. Primary hypothyroidism, seizures, because you can imagine you're stressing all the different kinds of neurotransmitters in the brain can cause prolactin to be elevated, and then a bunch of other causes listed here. A big thing is medications. And so when we're faced with a patient with a high prolactin, one of the first things we do is look at their medication list and see what's on it, because that's a very common cause of high prolactin that's not due to a tumor. And here's a long list. Basically, any kind of medication that interferes with dopamine signaling in the brain or other neurotransmitters. You can look back at the slide that Dr. Wardlaw showed you, showing you all the very complicated neurotransmitter inputs on dopamine secretion. And a lot of these, in particular, I'll mention the metoclopramide, used a lot for GI diseases, and all the antipsychotics. Tricyclics, very important causes of high prolactin. So always review the medication list and look up if it could be a cause of high prolactin. And then what are the clinical manifestations of high prolactin? As Dr. Wardlaw told you, the high prolactin feeds back and suppresses the gonadotropins at the level of the pituitary and hypothalamus. So basically, what you're dealing with is low levels of LH and FSH in the setting of high prolactin. So in the setting of high prolactin, you can have anything in the spectrum of hypogonadism. So high prolactin, the main clinical manifestation are the symptoms of hypogonadism. And we went through these already. I don't need to go through them, so we're running low on time. But patients can have anywhere from slight abnormalities of their menses to mild elevation of testosterone to really total infertility and no periods. So there's really a big spectrum of gonadal insufficiency due to hyperprolactinemia. And how do we treat hyperprolactinemia? When we're talking about a medication, obviously that's something different, but when we're talking about high prolactin due to a pituitary tumor, the therapy is our dopamine agonist medications. And this is really the one kind of pituitary tumor where we have great medical treatment. We have good medical treatment for other tumors, but this class of medications is great for pituitary tumors. And basically, there's a class of medications called dopamine agonists that are oral therapy that we give to patients with prolactin-producing tumors that suppresses prolactin production from the tumors, usually shrinks the tumor, and also in really 90% of patients, if tolerated, can treat this disease very successfully. Very rarely do we need to do surgery or radiation. And who do we need to treat? Well, you'd want to treat somebody with a big tumor, obviously, and big tumors can be shrunk very nicely with this medication also, but you also want to treat people with symptoms of high prolactin. Even in and of itself, the prolactin is not really harmful. You don't need to treat the number for no symptoms, but most patients will have some clinical manifestation that you want to treat them for. That's usually why they're coming to see you. So you're going to want to treat them. You always need to discuss all the pros and cons of treatment, but any patient with symptoms of gonadal dysfunction you would want to treat. And again, this slide just lists who you would want to treat. You want to restore the gonadal function by lowering the prolactin, getting the gonadal axis in line, improve sexual dysfunction, fertility. We didn't talk too much yet about galactorrhea, but a prominent symptom in patients with high prolactin, as you can imagine from what you heard about the physiology of high prolactin, is milk production in the breasts. It's very rare in men unless they're making a lot of estrogen, in particular if they're very overweight. But in women, especially women who've had children before and lactated before, galactorrhea is not uncommon. It's very common in patients who present with prolactinomas. So you would want to treat that for the galactorrhea, the milk from the breast, and also reduce the tumor size and restore the pituitary function. And normalize the prolactin level, also why you would want to treat somebody. And these are the dopamine agonists we have. Bromocriptine is just a daily medication. It's an older one, and cabergoline, which is a medication we can use once or twice a week. It's a very long-acting dopamine agonist. And then to end, I'm just going to show you how, and I think you probably have a case on this, how great it is when you can successfully treat somebody with a dopamine agonist. Because this is the before picture with the macroadenoma here, and this is the after picture after you've shrunk the pituitary tumor with the dopamine agonist. And this patient had normalization of the prolactin and restoration of their gonadal function with the dopamine agonist. I think that's it. Sorry to rush. Okay, thanks. If you have questions, come down.
But you're right. L-Dopa would do it, and certainly when you use those Parkinson's drugs, prolactin goes down. Yeah, and actually some of the Parkinson's drugs do work. It's just